Hi guys, welcome back to the channel. In this video, I will be covering the structure of eukaryotic cell topic of the A-level biology curriculum. So let's begin. So cell membrane. So that's a component of the cell, eukaryotic cell, which is present in every eukaryotic cell. So this is usually the most outside bit in most cells, but there are a few exceptions if they have a cell wall. So this is the cell membrane. So what they have is a phospholipid bilayer with embedded proteins. So for example, this is the cell membrane and it has embedded proteins, for example, the globular protein, and there's the glycoproteins, and there's a few other proteins. Right, so the cell membrane is selectively permeable. So what this means is it allows some substances to pass through the cell, whereas some won't be able to. So this allows the cell to keep a balance so the cell isn't harmed. And cell membrane is basically a barrier between the internal and external environment of cell. So if something is needed in the cell, it won't be able to pass out of the cell, and if something is not needed in the cell, it won't be able to go into the cell. So next is the nucleus. So nucleus controls the cell activity. So inside the nucleus there are chromosomes consisting of linear DNA. So because it's a eukaryotic cell, it will only have linear DNA. But if it was a prokaryotic cell, then it could have circular DNA. But in this case, it will have linear DNA, and it's bounded by proteins called histones. So we have the DNA there, and it'll be bounded by histones. And there are nuclear pores inside in the nucleus, so which allows the substances to move between the nucleus and cytoplasm. So for example, if we have, for example, a RNA, it will be able to pass through the nuclear pores in the nuclear cell, in the nucleus. So, and it has nucleolus inside the nucleus, which makes ribosomes, and it's made of proteins and ribosomal RNA. So that's inside the nucleus. So next is the mitochondria. So the mitochondria is the site of aerobic respiration, which produces ATP for energy release. And it's a double membrane. So the inner membrane is folded to form cristae, increasing surface area. So this is our mitochondria, and it has, you can see, this is the first membrane, and it has the second membrane. So it has two membranes, and the inner membrane, the second membrane, is folded, so increases surface area. And the matrix is basically the inside of the inner membrane, so this area. So the matrix contains the respiratory enzymes involved in the aerobic respiration. And finally, the mitochondria contains its own small circular DNA, so it can replicate itself. So inside the mitochondria, it has the DNA, so it does not need any other organelles, anything else to be able to replicate. It can replicate on its own. So chloroplast. So I know the animal cells do not have chloroplast, but plants and algae, plant cells and algae cells also come under the eukaryotic cells. So therefore, you can include chloroplast as a eukaryotic cell component. So the chloroplast is again a double membrane bound organelle. So like the mitochondria, it has one and two. So it has two membranes, and it has thylakoid discs which contain chlorophyll, and this is where the light dependent reaction occurs in photosynthesis. So these are one of the, this would be a thylakoid disc, and it contains chlorophyll, which is where the light dependent reaction of the photosynthesis occurs. And the thylakoid discs are stacked up to form grana. So, for example, in this case, these are stacked up and this forms a grana, and grana can be linked by lamellae. So this is a lamellae, and grana can be linked by it. And all of these things are located in the stroma. So inside the inner membrane of the chloroplast is where all of these would be included. And it contains, so like the mitochondria, it contains circular DNA and ribosomes, so it can make its own enzymes needed for photosynthesis and to be able to replicate itself. So it does not need any other cell or anything else to be able to replicate. Right, moving on is the Golgi apparatus. So it has flattened sacs called cisternae. So these are flattened and these are called cisternae, and this increases the surface area. So for example, it's not like this, it is like this, and this increases the surface area. So what it does is it modifies, processes proteins and lipids produced by the RER and SCR, so by the rough endoplasmic reticulum and the smooth endoplasmic reticulum, which we will be coming back to later. So for example, it would modify it or process it by adding carbohydrates to it. And it packages them into vesicles. So for example, this is a vesicle, so the modified proteins or lipids can be transported in and out of the cell, and this also makes lysosomes, which we will be coming on to next. So lysosomes are membrane vesicles produced by the Golgi apparatus, and it contains lysozymes, so zymes. So these are hydrolytic enzymes which breaks down or hydrolyzes pathogens or worn out cell components, and it's necessary in phagocytosis. So ribosomes. So ribosomes are made from one large subunit and one small subunit, and they do not contain a membrane. So there's no membrane in there, and it is free floating in the cytoplasm, or it's bound to the rough endoplasmic reticulum. So they're not in the nucleus, they're in the cytoplasm, which is outside the nucleus. And they're the site of protein synthesis, so they are where the proteins are made. So now the rough endoplasmic reticulum, or the RER. So this is a large network of membranes called cisternae. Again, cisternae, so like flattened sacs. So they are covered in ribosomes. So you'll be able to see, for example, this is a ribosome. So it contains a lot of ribosomes, and this is what makes it rough. So what it does is it folds the proteins from the secondary structure to the tertiary structure. So it would form bonds, and so it would turn them into a 3D structure, which is the tertiary structure. Okay, now the smooth endoplasmic reticulum. So if you're comparing them to the rough endoplasmic reticulum, so for example, this is the rough endoplasmic reticulum. It has a lot of dots, which are the ribosomes, but the smooth endoplasmic reticulum has none of these. So that is the difference when you're looking at a diagram. So these again are a large network of membranes, but these are without ribosomes this time. So what they do is they synthesize and they process lipids. So it's lipids, not proteins this time, which the RER does. So moving on to the cell wall. So even though the animal cells do not contain the cell wall, there are other eukaryotic cells, for example, plant cells, algae cells, and fungal cells, which do contain the cell wall. So the cell wall is the outside-most bit of the cell, and in plants and algae, it's made of cellulose, In murine, it's made of murine in bacteria. So even though bacteria isn't a eukaryotic cell, it's a prokaryotic cell. I just included it in there, so you know. So, and it's made of chitin in fungi. So what it does is it provides strength and rigidity to cell to withstand high pressure, and it prevents cell changing shape and bursting. So there can be changes in the osmotic pressure, changes in the water potential, which can lead to the cell bursting or changing shape, and so can harm the cell. But the cell wall, because it's strong, it has a lot of strength, it keeps that shape, so the cell doesn't burst. So now the cell vacuole. So this is plants. So this is the cell vacuole. So what it does is it contains the cell sap, which is basically a weak solution of sugar and salts, and it's surrounded by a membrane. So the cell vacuole has its own membrane, which is called the tonoplast. You do not need to know that. And it maintains the pressure in cell, so it stops the cell from wilting, and it stores and isolates unwanted chemicals in the cell, so such as things that can potentially harm the cell. It will just store them in there in the vacuole, so they're not exposed to the organelles in the cell, which could be harmful and deadly for the cell. So okay, so there is organization of specialized cells in complex multicellular organisms. So at the start we will have a specialized cell as the most basic structural subunit, and is specialized for a particular function. So for example, when we have stem cells, they're unspecialized, but when they're specialized, they will turn into certain cells. For example, a stem cell might specialize into red blood cells, so that will be a specialized cell. And then the specialized, a group of organized specialized cells forms a tissue when it joins, and they work together to perform a particular function. And then after that, organ. So that's a group of organized different tissues which are joined and work together to perform a particular function. And then after that is the organ system, which is a group of organized organs working together to perform a particular function. Right, so algal cells. So this is an algal cell. So what they do is they're able to photosynthesize because they contain a chloroplast, and they contain a flagella, for example this, and this allows it to move around, makes it easier to move around and to, for example, get nutrients or something. And it contains a cellulose cell wall, same as a plant cell, and it can be multicellular or unicellular. So now the fungal cells. So this is an example of a fungal cell. They can be unicellular or multicellular, and they contain a chitin cell wall. So their cell wall is made of chitin, and they do not photosynthesize as they do not contain chloroplast. So what they do is they gain nutrients from other things, as they can't make their own food, and they're made of long thin strands called hyphae. So hyphae, and these increase the surface area for absorption of ions. So as they're thin and long, they have a higher surface area, therefore they can absorb more ions. And the hyphae from branching networks called mycelia, which can further increase the surface area for fungal cells.